And with that, I would like to introduce Professor Phil Sharp, who will start the panel. Phil, are you there?
I'm there, thank you. This has been a fascinating conference. We've heard the cutting edge of how to use machine learning to identify combinations of drugs that could inhibit COVID, how to integrate that search into cellular gene expression, other data that is available, including the complexity of sequence in our drug structure and its impact on the cell. And we've heard many excellent talks, including how to design, and the last talk, antigens into peptides to vaccinate against COVID. So in a short time, actually, if you think about it, the COVID problem appeared in February, maybe early January. And in that interim time, there has just been an enormous progress with machine learning and prediction. And now we're at a stage where this is going to be tested in various validation models. And it is also setting a precedent for what the future of the field of pharmacology is going to be like. And that's what this panel will talk about. We have outstanding members of the panel. And what I will do is to basically call on them to introduce themselves. And then we'll have a discussion with some questions to each of them. And then we'll have a discussion among ourselves, and participants could also suggest questions on the Q&A part of the Zoom. So let's start with Ann Fisher, who is associated with DARPA. Ann, would you introduce yourself?
We can't hear you.
Program manager. Oh.
Hi.
We couldn't hear you at the beginning, Ann. Maybe you can start over. We still cannot hear you.
I can hear you now. Go ahead.
Okay.
No, I can't hear you. Can I?
Oh, now.
I can't hear you.
Yeah, we can hear now. Now we can, yeah.
Go ahead.
Okay. Earbud
Okay,
issues.
we
Sorry,
can hear
guys.
you. We can hear you. It's good.
Great. The technology agency that I work for is not responsible for the earbuds that weren't working just now. So I'm really happy to be here. Thanks, Phil, for the introduction and setting up the panel. As Phil said, my name is Ann Fisher. I'm a program manager in the Defense Sciences Office of the Defense Advanced Research Projects Agency, DARPA. Essentially, DARPA is a funding agency as part of the US federal government. And we look really to the far future of what technologies and capabilities we might need to help us sort of meet the need. It's a very high risk organization. And I'm really happy to be a part of this. Today, we have several efforts that really are about accelerating the way that we can do discovery across the chemical sciences. I come at this as an experimentalist, as a chemist, and I've really learned a lot over the past several years about AI and machine learning and how those two fields can really play together and play off of each other to advance pharmaceuticals as well as other technologies across the chemical sciences.
Thank you. And we obviously will come back with questions and discussions, but moving on to introducing the rest of the panel. Nobar Afei, can you introduce yourself?
For some reason, we cannot find him. So we're trying to find Nobar. So while Ignacio does the finding, Joe, maybe Najat can introduce yourself.
So let's go on into Najat Khan, who is with J&J. Nice to see you.
Nice to see you again, Phil, and thank you, Regina. by the way, the last talk was great, so looking forward to more discussions on that. So introduction, so I am the Chief Data Science Officer for Janssen R&D. So Janssen is the pharma arm of J&J. I am also the Global Head of Strategy and Operations for Janssen R&D. So in my roles, just the bottom line is I focus on what is our pipeline, what are the medicines that we're developing, both in discovery and development, and then very deliberately applying data science across that entire pipeline to either make medicines in a more effective way or in a more efficient way. My background, confluence of three things, computational science. I did my PhD in organic chemistry, so lots of time spent in the lab, and I loved it, especially focused more in the oncolytic therapeutic space, and then a lot of time, over 10 years or so in the industry, just understanding both R&D, commercial, really around how do you drive decision-making from a asset medicine perspective, access to patients, development, building on those capabilities, both the computational and the science and medicine. And I think, Phil, maybe one thing just in tied to this panel, my team, my data science team has been working really closely along with our clinical group and then also our operations group on how do we use AI machine learning to deploy and accelerate the discovery and development of our vaccine programs. So happy to speak to that as well later.
Thank you. And the next fourth member, if Nobar is on, is Regina, who's been with us since the beginning of the conference. And Regina, I'm not sure you need to introduce yourself.
Exactly, I don't, totally not. I will let Nobar introduce himself.
Is Nobar on the?
Yeah.
I'm on. Phil, I'm on. I could hear you guys. I've been watching for the last hour with great interest, but apparently on the webinar link and not on this link. So I've been upgraded. Thank you. My name is Nubar Fan. I am CEO of Flagship Pioneering and also co-founder and chairman of Moderna. Flagship, for those of you who have not heard about us, is an enterprise that is engaged in making pioneering innovations and creating companies that can advance the products that come from these innovations. These are all life science-based platforms, primarily going after health and sustainability. And we have a number of projects that involve applications of machine learning and AI in the pharmaceutical kind of value chain in general, and in particular with respect to COVID as well.
Thank you. Let's start with a few questions. Anne, we're immersed in vast amounts of data related to COVID, and predictions of the future are obviously very important to us because speed in dealing with this pandemic is everything because of its impact on the economy. How does DARPA look at or think about integration of data, how to use data, and how does DARPA promote the sharing of data, which is obviously something that is a major issue in moving rapidly forward?
Yeah, absolutely. So I think I'd start by saying, I think drowning in data sort of depends on your perspective. I think Regina might argue the point that we don't have enough data. And what again, coming at this from the sort of experimental perspective, what I'm trying to do is think about how we can develop or exploit the right data for the question that we're asking. I think Regina started off the conference talking about data sharing, which is part of your question, the ability to exploit existing data. And I think critically, we also need to think about how we can generate new data sets rapidly. So for COVID, that might be about screening assays, which maybe one could argue are in sort of the line of high throughput, as long as you have the compounds available. So we're limiting ourselves from my perspective to that. Right now, if you want to talk about developing new compounds, obviously you have to think about regulatory aspects. But what we're trying to do is really think about this from the framework of what tools and technologies do we want, in sort of a future of being able to address the question of closed loop design of molecules to address certain questions or to have certain properties. And I think critically there, the ability to exploit existing data, to be able to reproduce that data quickly, and to be able to then learn how to exploit, those data sets from the perspective of machine learning, really gets at all of the types of things that people were talking about today: representations, reproducibility. All those aspects are huge components. So I think we not only need to be thinking about today in the pandemic and how we use the tools that we have today to address therapeutic development, both antivirals and antibiotics, but also what do we want to be building for the future when this happens again, right? And that's really where we are, sort of thinking through how we can build those capabilities today. Now, a brief comment about data sharing. The programs that I run are all open source. So all the data sets are public as they should be. All of the tools and technologies that we're developing, code, experimental systems, published on GitHub. And that's really where, this is government-funded work for the benefit of society. From my perspective, we should be sharing as much as possible.
Wonderful. I'm going to ask you a question, or just make a comment, and if you have comments or questions about it. Many years ago, there was other coronavirus, infections, MERS, SARS-1 and others. A lot of data has been collected over time. Those viruses will be similar to COVID-19. But again, even with those data in the books in many places, it was very difficult to access that data and then to be able to take that historical data. Does DARPA look back and in any way can DARPA promote the sharing of that type of data?
I think it sort of depends on who generated the data in the first place, right? What we can do is from time t equals 0, which I consider, when I started the program called Accelerated Molecular Discovery, is mandate that as part of the awards themselves. We're building databases. We're trying to understand how, even how computers sort of understand the data sets that are being generated, how we can tag them appropriately, those types of things. I think those are all still relatively open questions that the community really needs to come to some sort of agreement. We'll never agree on everything, right? No. But something where we have an accessibility that's embedded in the data that we're generating, I think is critical.
Thank you. Nadja, as we've seen, data does promote the advancement in this field. How is Janssen using data to, accelerate drug development or vaccines? Or, what's the nature of your effort? How do you visualize it?
And Phil, is it more for a COVID vaccine or just broadly? Because I can speak to both.
Both drug and COVID.
Okay. Yeah, I'm just maybe taking a step back. One of the things we're doing with data science is we're looking at the use of all of these different data sets that you mentioned, both retrospective data, real world data, and of course, prospective data as well, both in discovery and development. So in discovery, there are the age-old questions of what's the right target that we should be focusing on. And there were some good talks here as well. And then also in terms of how do you determine what's a high quality NME? How do you reduce that attrition? So we're using techniques like cell painting that Ann Carpenter has pioneered to really understand, just looking at a cell, just looking at images, can you predict what the molecular properties will be? And we've done it across quite a few programs and are actually scaling it across the board for our small molecule programs over the next year. I think as a former chemist, there's a huge amount of value if we can make more intelligent decisions early on in a more parallel fashion versus all of the bespoke assays that's done today. I would say on the development side, the questions look different. So, there's a huge question around whether it's rare diseases, new indications, COVID, what are the right endpoints we should be looking at? A lot of the times We look at some composite endpoints that are good for clinical trial purposes, but pivoting more towards real world based endpoints helps you both with regulatory approval, but also access. And, that's starting to fold more and more together. And then I would also say, and that ties to other aspects such as external control arms or synthetic control arms to really contextualize what we're doing. Take COVID as an example, for instance. With all of these vaccine trials and all of these therapeutic programs ongoing, wouldn't it be useful to actually think about, can you mine the real world database to actually think about controlled placebo arms instead of running one for each? We are not there yet. And it ties to a question you asked, Anne, which is what I would love to see is a more linked, interlinked database in the US so you can actually use that. Today, the standards are different. It's fragmented, very hard to do, very challenging. And with COVID on top of that, Phil, the standard of care has changed so much, right? Every month. So how do you even do that? It's not contemporaneous. And then the last thing I'll say in the development space is around operations. Running of a trial, recruiting patients, finding patients. We use a lot of real world data, especially near time, to actually be able to say, where are the eligible patients located? Be identified, of course, and then be much more targeted in the HCPs we go to. We're already doing this. And the one thing that I really like about it, other than being faster and more targeted, is we see much more diverse patient pools, because there are a lot of them are in community clinics that we usually don't go to. So if we talk about diversity, let's not just talk about reactively what we can do, but let's actually, before we even select sites, ensure that we have a good balance going in. And I would say on the operations side, as an example for COVID, we've done a ton, my team has done a lot of work actually with Dr. Dimitri Bertimus from MIT to be able to predict where these hotspots are going to be. Because one thing with any of the vaccine programs is you want to be in areas of high transmission. It helps you with the event rates, faster trials, etc. And we build a really complex, I would say, machine learning model that predicted about 3.5 months ago that we needed to be in certain areas in the Midwest or, LATAM or even Europe. And that uses, a lot of the data that you'll see in Johns Hopkins, right? The infections and the numbers there, but also other things like mobility data, social compliance, disease severity and how that's evolving. So I just think combining all of the qualitative and quantitative factors and diversity elements, social economic status, really, and we had very high accuracy with these models, over 85%. So that's really helped us hone in on where we go for our hotspots as well. So I think across the board, there's many areas we're using data science, both data discovery and development.
Thank you. I'm going to come back later to talk about combinations of drugs, because I think combinations actually present for a pharmaceutical approach de novo major challenges. I think it's a topic worth discussing given the conference. Nobar, how is Moderna incorporated machine learning? How do you think about using AI in production of a vaccine? We heard a very interesting talk at the end about T antigen immunity. T cellular immunity and peptides and MHC. Speak a little bit about your thoughts about vaccine development.
Sure, Phil. And maybe what I'll do is I'll answer it initially centered around Moderna, but maybe I'll just zoom out a little bit because there's plenty of other places where within our ecosystem where these kinds of tools are being heavily deployed. I think within the context of Moderna, as was discussed by Professor Gifford, Moderna used the spike protein as the preferred immunogen and used an mRNA construct and worked rather on the rapid deployment of that as a drug, as a vaccine in this case, but the formulation, etc., as opposed to trying to figure out which subsets of peptides could be used and augmented. The thinking being that if we could get a vaccine demonstrated to work, we could always do additional versions of it, which we will in the fullness of time. I will say that Moderna heavily uses data because its platform, the RNA platform, is innately a data platform. And we're basically putting code into a subject and expecting the protein, the corresponding protein, to then run the program. And so one of the things that I'll say proximal to the work we've done in the COVID vaccine and the predecessor to it was our cancer vaccine work, where we have an active a trial with individualized tumor sequence derived multiplicity of antigens, in some cases 30, 40 different antigens that are heavily derived from data and algorithms to select these peptides, and then turned around and reintroduced to that patient within a couple of weeks. It is actually that process that we already had running and in clinical trials that allowed Moderna to very rapidly turn around and make yet another antigen, in this case a spike protein. But the data infrastructure and the antigen selection that we do in cancer vaccines is a very advanced capability. And as David said, might well make sense to apply that not just to cancer, but now back to viral antigens. But just to zoom out a bit, I'll also mention that simultaneously in a different project that's called Generate Biomedicine, we've deployed machine learning approaches through which we've been able to, over the last year and a half, develop the capability to computationally specify antibodies for completely de novo antigens. And within weeks of having the sequence, had generated hundreds of antibodies, which after one cycle allows us to do purely computational learned affinity maturation, and showed that we could actually make quite specific, sometimes general in terms of binding multiple spike protein variants, but sometimes very specific ones. And so I think the future of antibody design and specification will and is being heavily advanced by data around initial sets that then lead to optimization. A couple of other things I'll mention. We're using heavily the same kind of data approach, but now for cells with single cell RNA sequencing, particularly to be able to track cell behavior changes in the context of disease, including viral infections. Cells respond in a particular way. They go through a trajectory, and understanding that actually reveals a set of targets that would otherwise not be seen through traditional target validation methods. And then finally, along the lines of what Professor Gifford mentioned, we have another project called Repertoire Immune Medicines that is literally for not just COVID, although we started working on that right away as well, but for multiple viral infections, actually doing full-on sequencing of every possible peptide with the TCRs to create synapse maps, basically, from which you can then look at what is prevalent in the population of immune responders. And so we did a lot of work on that with CMV, and now have been training it to. So there's a lot of different approaches. Of course, when the shutdowns happened, everybody basically decided to use their wares, just like at MIT labs that I'm very familiar with, on COVID. And it's been a really interesting time because there's an unprecedented amount of data and publicly available data that's been spewing out in the last 6 months. And I'd hate to see that kind of be that genie put back in the bottle so that we can only imagine what that world used to look like or remember. I really hope that this will be an irreversible change.
Wonderful. Do you use multiple RNAs with multiple spike protein to produce vaccines, or do you model your vaccination on one spike protein?
Phil, because the priority in that case was a single vaccine that could be tested in humans as soon as possible, it was actually piggybacking off of work that had been done at the NIH and then also in partnership with Moderna for a year earlier in MERS that was getting ready to go into clinical trials. And that knowledge about the spike protein and the mutations that you need to make to be able to make an immunogenic version presented, that was the thing that we decided to lock in on. We have made many other constructs, of course, from a research standpoint to see if there's variations in animals. And so we have a lot of know-how that's been developed there. But there, and also oddly enough, I mentioned in the cancer case, there's 35 antigens that are in a single construct that are all translating different peptides. So you can, it's pretty flexible.
Good. Regina, what did you take away from this conference?
From the conference, I was very much, of course, aware of what we are doing at MIT in terms of, developing antivirals. And I saw the same trend that it seems to be an obvious place really to do very tight modeling of biology and chemistry. So Anne, our program manager, knows that we started our journey really looking at chemical structure and making predictions from chemical structure. And in many cases, it actually worked pretty well. And I was naive enough to claim that we don't really need to know more because we have screening data and we can predict. But what we've discovered here that, it seems that in Nubar's world, there was a lot of data. In our world, there much less data. And even the data that was available was very contradictory, very noisy. So introducing biology and thinking in novel ways how to incorporate it was really an eye-opening moment for me. And as we've seen in some of the talks, not all, but some of the talks, that in order to do this integration, you really need to have very novel machine learning algorithms. You cannot just take what is out there at Apply because you would not go very far. So on the exciting part, we have a big space on the machine learning side to do the innovation. And as you said earlier, you've noticed in our work in the group by people in Miller Group and others that a combination became really an important topic, which was unaddressed until now. So it kind of, I think that it brought us, this COVID crisis brought us closer to real discovery, reality in discovery. And it seems to me that many groups at the same time getting similar realizations.
I particularly was excited about how you're integrating cellular and molecular information and mechanism into discovery. There were several talks about that, but as that becomes more common, it will also feed back and tell us more about cellular systems and how to interrogate those in an organic way, as well as having byproduct or the direct product of having better drugs and more effective drugs and new drugs. Najat, we were talking about drug combinations. How do you view development of drug combinations? It's a complicated course of clinical development and other developments. Can you speak to that?
Yeah, no, I can definitely speak to that. I mean, just in terms of drug combinations, it looks different in different areas and indications as well. So for instance, of course, we're looking at it in immunology. So a lot of the IBD diseases, I mean, there's a lot of therapies out there, but there's a lot of relapse on those therapies. So how do we think about the right drug combination? For me, it starts around being able to identify the right subpopulation that would actually respond. And now it's like, take Crohn's as an example. That's an area we're actively looking at. The holy grail there would be to really understand how to stratify those patient subgroups. And while it's easy to say, one of the challenges, Phil, is just having data and how the data has been collected. Do you have all the right parameters that you need? Now, in one way, we have a lot of internal data from prior trials that have been run, assays that have been done, biomarker studies, etc. There's also a lot of real world data, but back to Regina's point, it can be noisy sometimes, because the missingness of data, the fact that it's not integrated across different health systems, the fact that there are gaps, right? And also the adherence can be variable is challenging. But what we are doing, it is an area that we're actively working in. So we have new MOAs such as the NKG2D4500. It's a new mechanism of action for Crohn's disease. And we're looking at, okay, what are the right combination therapies there that would actually target the right subpopulation? But the question starts with the right subpopulation. And that's where data science comes in, because you use a lot of the clinical trial data, prior clinical trial data, and real world data, and biobank data wherever we have it, to be able to identify those and stratify those pools. Now, if I think about oncology, it's a very different place in terms of how do we think about combinations, right? And I wouldn't say it's easier at all. None of this is easy, but some of the endpoints that we're looking at, again, to be able to do that subpopulation that I was mentioning, is a little bit different. The endpoints are a bit harder, hard endpoints versus soft composite endpoints. So you can actually look at both your clinical trial data and real world data to be able to parse that out. So I think, Phil, for me, of course it's necessary, but it has to be, we first have to have a hypothesis or a thesis in terms of which populations it could impact more, versus what's been done sometimes is let's just combine different drugs and see what the effect size is. And especially in large trials like in immunology, like in IBD and others, I don't think it has been productive. Maybe to show some gain, but from an access perspective, there's significant challenges. So that's how we're thinking about it, and we're applying a lot of data science, machine learning capabilities to do that.
Wonderful. I don't want to dominate the panel talking to each other, so please feel free to inject yourself with a question to someone else or a comment to someone else. But I will move on to Anne if I can.
Phil, can I ask a question?
Sure,
Jad and Nubar, as a homeschooling mom, so when are we going to have a vaccine available?
I'll let Nubar go first.
Yeah, well, I think what we've publicly said is that we expect by the latter part of November to be able to have sufficient safety data based on what the FDA has indicated, which is 2 months of data on half of the population in the trial having received 2 doses, and the possibility of an early read on efficacy if in fact the vaccine is efficacious enough to show statistical significance at an early read. If that doesn't happen, then that should happen at the 150 event level soon thereafter. And so we're talking about kind of probably a few weeks of difference. Now thereafter, there's a lot of kind of confusion and uncertainty about what happens if there is efficacy and safety demonstrated. You're all hearing, because it's on every television channel and even the sports channels are speculating about vaccines and the like, that at some point, the FDA and other regulators will have to decide if they want to make vaccines available through emergency use authorization, and in which case to whom. And then eventually that should lead to BLA filings. And that eventually, I think, is the general notion has been in the late Q1, Q2 timeframe, just based on how long it takes for the trials to complete and the data to be aggregated. And then I think there's a question of supply. Yeah. And I think public statements have been made by several of the manufacturers that, while they expect some amount of supply before the end of this year, which is kind of in the tens of millions of doses, if it's even allowed, that it'll be kind of towards the second quarter of next year when supply should ramp up. Hopefully multiple vaccines will get approved so that we have the supply of multiple. There's a general misunderstanding that no matter how many times we say people tune out on, which is that I don't think any of the vaccine testers are racing against each other, because we all recognize that the sum total of all of the supply we can make is far less than what the world will need over the next 12, 18 months. Necessarily, once all that supply is online, it's going to be too much supply if everything gets approved, but we'd rather take that risk than the other risk. So I think everybody hopes that multiple vaccines make it. And then, as was mentioned earlier, and again, Professor Giffords commented on this, we'll start looking at whether vaccines are working differently. They're working differently on different aged people. And I think that will also dictate distribution considerations and uptake considerations. But that's kind of the general thinking. Of course, there's a chorus of people, some of them CEOs of large pharma companies, who have stated unequivocally that any vaccine that takes less than 3 or 5 years to develop is inherently unsafe. We don't believe that. I think everybody's doing everything they can to follow the rigor of science. But nevertheless, there's going to continue to be a lot of the uptake rate, Regina, I cannot tell. I think it could be a very, very flat kind of uptake rate as people gain confidence, or it could be different. So I hope that's useful.
Thank you.
Yeah, I'll agree with Nubar. I mean, I think the bottom line, Regina, for you is probably going to be, I'm thinking more like the June, July timeframe, right? Regardless of what works versus not, the distribution and who do we, vaccinate first, all the points that Nubar mentioned is right. I completely want to underscore what Nubar said, which is having multiple options in this case is a good thing. And the fact that we have had, across different companies, some pauses is a reflection of the fact that nobody's compromising safety, right? So I just hope that that's something that will become more and more well understood over the next few months. But yes, probably next summer. I think the winter, you'll be homeschooling your son.
Thank you.
And Phil, since you encouraged us to comment among each other, I think the other thing probably worth to keep in mind, again, I found the prior presentation very intriguing and provocative, but I think what it speaks to and what all of us realize is that we really don't know how the immune system will respond to this and other such viruses. It's a shame that vaccine research has been so underfunded and vaccine development has been thought to be kind of a long process. Therefore, nobody really wants to engage in it. But I think there are as many questions about the significance, let me just tell you, on significance of T-cell response in infectious disease virus among the experts in the field, as there are questions about the absence of T-cell response in vaccine developers. I can tell you many vaccine developers have a ton of data about the relative importance of these. We just don't know. And of course, the fact that we don't know to scientists should raise questions, but to politicians raises concerns. It's the same phenomenon, but different words. And when it's concerns, everybody go, I don't know how long it's gonna last. I don't know if the antibody's gonna stick around. I don't know. And I think that kind of non-scientific approach is gonna give way over time to multiple scientific experiments. And then we'll look back and say, probably during COVID is when we figured out how the various components of the immune system have to be activated to create the best possible protection. And that we're going to learn over the next multiple years, not in the next 6 to 12 months.
And just building on something that Nubar mentioned, if we think about if this happens again, how do we respond? And how did we not respond? How it wasn't perfect this time? I think there are two things to keep in mind. One ties to that vaccine, which Nubar, you're mentioning. I mean, we know there are other coronaviruses. I mean, there's been a lot that's been published, early work and bats and other things. Being able to predict what that next strain would be, what that epitope or antigen would be, I think would be of huge value to then be able to better understand, the T-cell response and therapeutics that you might need. There's not that much work being done today, and I think it might be a catalyst, but I think there is a whole host that we could even look at and say, okay, of this 50, are there 5 that we predict that could have a higher likelihood? So I think that's one, and that could leverage some of the prior data, Phil, you were mentioning. And then the second piece, and the first is more about being preemptive, right, as to how do you predict it. And the second is more around what do you do once there is a pandemic? I mean, I can't underscore how long it took for us to really even understand what's happening on the ground, just getting real world EHR, claims, whatever data. It took a long time. There was a little bit better, there was variation in the standard of care. We could try to normalize it across. And that's because data is so fragmented, right? Even if you look at a lot of the EHR claims data sets, you would think that real time, if you can look at that and really understand outcomes, that would be a huge use, right? Like I mentioned, the standard of care is changing. You can try to do external control arms, all of these things. Think about if there's an EUA, what do you do with the patient, with the subjects that are in the placebo arm? How do you follow the subjects for a long period of time? Passively, right? That is something you can do using EHR data, but there's a construct, almost as foundation, that's not where it needs to be. There have been some alliances in terms of sharing of data, COVID-related data, and also endpoints, et cetera, that's happened. But I do hope that that's something that continues. A lot of it can be pre-competitive, I think, that it actually continues and it doesn't go back. And I think that's another point Nubar mentioned. So those two, one is just the research, but the other is also how do we get the foundation right so that we can actually learn and do better surveillance and react faster.
We're at a disadvantage with a fragmented healthcare system, as you well know, but it also is a system that responds quickly. And do you want to comment on the vaccine development issue?
So I don't have any comment on vaccine development. All my work is focused on small molecule therapeutics. But I think I would make a comment here. It kind of echoes what Regina was talking about, and it sort of looks at two ends of the spectrum of what we're discussing on this panel, which is critically important to find something that helps us today. And there are so many great minds racing to do that. And as the colleagues on the panel have already said. And the other side of it is, from my perspective, and this is where, you know, sort of we're steeped in our day-to-day, is really trying to help understand what matters most when it comes to being able to predict a molecule with a particular activity. So Regina talked about how she realized now that we need to incorporate the biology. We need to incorporate dynamics. We need to incorporate, you know, there some computational modeling that the folks have talked about today that we can leverage to do transfer learning. But the reality is we don't actually understand, just like we don't understand on the vaccine development side, we don't understand what features of molecules matter in each particular case for each particular mechanism of action. And I don't think, I'm not naive enough to think we'll always understand it to sort of the nth level that we need to. But the question is, how do we find the most efficient approaches to this development? And that's really, I think, what we're focused on from the perspective of therapeutics development, is understanding, you know, the science around it, so that we can then accelerate the development and help colleagues such as the folks here on the panel from the development effort.
I would just say we started this conference with a discussion of epitope drift in virus. You do not want to underestimate if this virus can mutate to a new antigen spectra on the spike, that we may see multiple generations of COVID with its lethality moving through the population. I don't have data, nor do I know of anyone that has data, but I think it's something that we need multiple approaches to continue to ensure we don't have that problem, and we're more prepared for future COVID.
Sorry, yeah. Phil, can I just ask one question to Anne directly? So, because we were interacting very closely with Anne on our, you know, we made prediction about single compounds. I think in June, we just yesterday got the response. Luckily, 2 compounds were active. We still are working on the combination. And to me, the fact is that, you know, you have computational folks who don't have the data to train. When they finally somehow get the data, they cannot test it. And there is this huge disconnect between people who generate data, who understand biology, and people who actually, you know, are capable of developing new models. It's really a serious concern. And also experimentally testing it. So, and I'm sure that MIT by no means is unique in this situation. There are lots of other folks around who want to contribute, who are talented machine learning people, but due to all this limitation cannot really jump in. So my question is, like, in terms of the, you know, government view and the priorities, what is the main lesson about kind of building infrastructures that next time, hopefully there will be no next time, but even this time, that we actually can make it much faster and effective, you know, this kind of learning and trial and eventually delivering the treatment?
Yeah, thanks, Rena. It's a great and complicated question. So I think I'll say a couple of things. One is that it's very clear when we think about our domains and the language that we use in our domains and our understanding of other domains, we all need to do a better job communicating across the area. And what I've found over and over and over again, I have programs that have chemists and engineers and mathematicians and you know, computational folks, and really you have to spend a lot of time getting everyone on the same page. But when you do that, it becomes, you know, the results are sort of amplified significantly, because it becomes much, much faster and much easier to address those concerns. And I think you're a great example of this. The team at MIT, who in the precursor Make It program really, you know, started to learn about this crosstalk. And so it's hard to think about how you might do this at a more global level. But I think that it's critical when we think about education, training in graduate school, postdoctoral programs, to really have that variety, have chemists who can code. I'm an experimentalist, never coded in my life, right? But now I don't have to code necessarily to be able to talk to someone who does and understand that language. And it's sort of a non-answer, but it's because I don't know how to solve the problem other than to continue to get on sort of my pulpit and talk about how we all need to be in the same room. And we all need to be sort of clearly and openly communicating with each other, which you've heard me say on the phone before. And again, you sort of, sometimes we just have to knock our heads against the wall for a while, and we break through eventually. And I think we just have to continue to do that. And I think the community generally really has to recognize that. And I think we are. I think, you know, there's a lot of evidence that folks are getting outside of these sort of stovepipe disciplines. And I think we just need to continue to encourage that as we move forward.
Thank you.
We're approaching the time we will need to close. I want to thank all the panelists. It's been a very interesting discussion, very illuminating. And I'll give Regina, she started the conference and she can close the conference. Regina, you have the last word.
Wow, thank you. First of all, it's the first time that I see Phil in informal clothes, so it was worth to do.
Oh, yes.
For that, correct? But at any rate, thank you very much for all the attendees, for the panelists, and for our speakers. And I just would like to say that we recorded the materials. We will check with the attendees if they are okay with us releasing the data, releasing the recordings. We'll put them all online, and we will continue to try to make our science public. As Anne highlighted, it's important to bring everybody together. And just follow us on the JClinic website and social media. We'll announce our next event soon. So thank you very much and have a great day. Bye.
Thank you.
Bye.